Hey guys, and welcome to this lecture on the cell and cell transport.
All right, a few cell fun facts for you. Cells are the smallest living unit. There are over 200 types of cells. There are trillions in our bodies. The shape reflects its function. We're going to talk more about that whenever we get to tissues, really, or histology in chapter 4. And cells are mostly carbon, hydrogen, nitrogen, and oxygen. And then there's a link here for you that you can click on if you go to your PowerPoints. All right, cell theory. So let's start by talking about the time when you were 6, 7. Is that when around the time you start to ride a bike? Anyways, let's say you are riding your bike and you probably were like me. You weren't very successful the first time you did it. You fall off and you scrape your knee. And the first thing that happened was your mom put you in the car and she ran you down to Walmart. And she started looking for aisle 13 because she knew you were going to need some more cells. And Walmart has everything you can buy, right? So she went to aisle 13 and she bought the cells, they go for, like about $10 a pack. So you just, pick up a pack of cells. Usually there's about 100 cells in a pack, around $10 a pack, And then she took those cells and she patched up your wound. Everybody remember that? No, because that's not how it works, right? Cells come from other cells, thank goodness, right? So maybe she put some Neosporin on there and to make sure you didn't get a bacterial infection. And she probably put a Band-Aid on there. And in a few weeks, it probably looked as good as new. You may have had a little scarring, but you'd form a scab and the scab would fall off, and then you were back to normal. And that's because cells come from other cells. Cells do not come from aisle 13 at Walmart, okay? While Walmart does have almost everything you could need, it does not have cells. So cells come from other cells is one of the three main points to cell theory. Cells are the building blocks of life, and a single cell is the smallest unit of life. Cells differentiate. So here you can see the sperm, and it's fertilizing that egg. And then we start cell division. And as this cell division occurs, these cells divide and then they specialize, which is what cell differentiation really means. Divide and specialize. And so these, from the single fertilized egg, you form epithelial tissue, connective tissue, muscle tissue, and neural tissue. And each of these different types of tissues performs a different function. So these cells are so specialized that they perform a different function from each other, which I just think it's pretty crazy how you can go from this one cell to having a heart and having a liver and having a pair of lungs. And the cells that make up those tissues, that make up those organs, are just so different from each other in order to perform their certain function.
On this slide, we're going to talk about the parts of the cell, also known as organelles. So let's start with the cell membrane. That's the outermost portion of the cell. It's going to determine what enters and leaves the cell. It's basically the barrier between the inside portion of the cell and what we call the extracellular matrix or the cell's environment. Then we have the cytoskeleton. The cytoskeleton is going to function much like the skeleton of a human. It is made up of little protein fibers, and basically these protein fibers help to give the cell structure and support, because animal cells do not have cell walls. So this is important to know. Animal cells do not have cell walls. But when we looked at photosynthesis that occurs inside the chloroplasts of plant cells do have cell walls. So your cells and my cells do not have cell walls, which means they're not as structured. They don't have as much support because we're missing that cell wall. So the cytoskeleton is basically a support mechanism from within the cell. If you look around at all of the yellow portion here, so all the yellow, you can actually see what's called the cytoplasm. So this is the cytoplasm. I'm going to add it right here. Whoops. My pen has gone crazy. Anyways, so the yellow portion is the cytoplasm. It is a jelly-like substance, and it surrounds and also holds the organelles. Then we have the nucleus. The nucleus is typically in the center of the cell. Sometimes it's flattened, sometimes it's nice and round. Just depends on which cell type we have. And the nucleus, it houses the DNA and genetic material. It provides instructions for building proteins. And proteins are going to be very, very important in the body. They do almost everything. One example of a protein that's really, really important that we would need is hemoglobin, and that's going to transport oxygen on our red blood cells. Our tissues are going to need oxygen so that they can do cellular respiration. Which brings me to my next organelle, and probably my favorite, which is a mitochondrion or a mitochondria. So here you can see they're almost like bean-shaped structures, and the mitochondria make ATP. Once again, my pen has gone crazy, but anyways, they make ATP or energy, and they do that via cellular respiration. And if you remember what cellular respiration is, it's glucose plus oxygen through the process of enzymes gives us CO2, ATP, which is our goal, and water. So the mitochondria is very important. Basically, without this energy, the cell cannot function. So here you can see there's more than one mitochondria in this particular cell, and depending on what the cell is made to do, what its function is, depends on really the amount of mitochondria that's going to be found within it. For example, neurons, which are brain cells, those are going to be, they're found in the brain and the spinal cord. Let me rephrase that. They are going to be heavily basically you're going to find a lot of mitochondria there. I don't know what I was doing there. Anyways, so basically you're going to find a lot of mitochondria in those cells because they're going to have to function to send electrical impulses and signals. Also, muscle cells are going to need a lot of energy to contract, so you'll find mitochondria in muscle cells as well, especially the heart, cardiac tissue. Then we have ribosomes. Let's talk about the ribosomes. So you can have free ribosomes, which are the little dots out here. I don't even know why I use this pen, in the cytoplasm. And then you can also see all of these little dots here, which are also ribosomes, and they're associated with that purple structure called the rough ER. So here you see you have the endoplasmic reticulum, and it's separated into the rough ER, which has the ribosomes, and the smooth ER, which does not here. The ribosomes make the proteins, so that's their job, protein synthesis, and synthesis is just a fancy term for make. So these ribosomes make proteins, and the rough ER, its job is to store those proteins. It'll help fold the proteins as well. And then the smooth ER is actually really important for making lipids. You're going to see why lipids are important to the cell. But the smooth ER, it stores and produces lipids. So that's the function of it. Let's move on to the Golgi apparatus. Up here, it's this dark purple structure, the Golgi apparatus. Oops. Its function is to package the proteins and the lipids, and it packages them into these little vesicles. If you see these little vesicles here, so it'll package them in vesicles, and the vesicles can either move the cells or transport the cells, transport the vesicles. These vesicles can transport substances around the cell, or you can actually see it here happening. They can actually transport molecules outside the cell. Then we have the lysosome. This is also one of my favorites. So when you think lysosome, you should think Lysol. I'll write that out here. So I think of like a clean bathroom, and basically the lysosomes are the cleaners of the cell. They clean up or engulf and get rid of damaged organelles and also pathogens that might enter the cell. And then lastly, we have the peroxisomes. I'm not sure how much your book talks about these, but basically they help to digest fatty acids, which is a type of lipid. So hopefully you feel pretty good with the parts of the cell or organelles and what they do. Make sure you feel really comfortable with these for your exam. All right, so I want to focus in on the cell membrane. It's really, really important part of the cell. Like I said, it determines what enters and leaves, but I want to talk to you more about what the cell membrane looks like. So it's made up of what's called a phospholipid bilayer. Bilayer meaning two. So it's a phospholipid bilayer. The bi there means two. So over here you can see two layers of the phospholipids. Each phospholipid looks like this. It has a hydrophilic head. Hydrophilic means water loving. And hydrophobic, which is the tails here, these are the fatty acid tails, means water fearing. And you can see how the tails actually are much longer than the hydrophilic head, so they're much bigger as far as the phospholipid goes, which is going to be important when we look down here in this diagram. So we have the watery cytoplasm. Remember, that's going to be found on the inside of the cell. We have the watery extracellular fluid that's on the outside, and this phospholipid bilayer here known as the cell membrane. And it's these hydrophobic tails that actually keep the water from the outside, from just freely moving in and expanding or bursting the cell. And it also separates the water or the cytoplasm that's inside the cell from the extracellular fluid that's on the outside. And this water isn't going to be able to move so freely. It's actually going to need some help, and we'll talk about that in a little bit. But I want to make sure you feel comfortable with the phospholipid bilayer. What does a phospholipid look like? It's right here. It has a polar or hydrophilic head, which means it's water loving, and it has two fatty acid tails there, which are water fearing or hydrophobic. You should know what hydrophobic means. All right, so it's kind of hard to believe, but the cell membrane is actually fluid. It's called the fluid mosaic model, and basically it is actually moving. So it moves, which is important to allow substances to move in and out of the cell. There are certain parts of the cell membrane that I also want to show you here. So you can see these glycoproteins and glycolipids, and basically these are both carbohydrates. And they're important in cell recognition. And I like to think of these as like the cell's flags. So if you were going to put up a flag, okay, maybe you have a castle or house or something, you're not going to put the flag typically on the inside, right? You want to show everyone your flag, make it be known. For example, my fiance is really excited about moving here, but he has asked that we put a Scottish flag outside our home. So that's fine with me, but he's proud of where he comes from, and so he wants to fly his flag. These glycoproteins and glycolipids are basically cell flags, but they are more or maybe it's like a bumper sticker on your car. And maybe you see somebody with the same bumper sticker as you, and you're like, oh yeah, hey, how's it going? You have something in common, right? You can recognize each other maybe that way. So that's what these are for. They're important in cell recognition, and they're located on the outside of the cell membrane. Then we have these cholesterol molecules. If you look here, these yellow structures here, the cholesterol molecules are going to be really important for stability and helping to keep the cell membrane fluid. So cholesterol helps with stability and keeping the membrane fluid. You also have protein channels. So remember how I said that not everything like water just gets to move freely into the cell membrane? Well, for substances like that, they have to go through protein channels. And sometimes we have protein channels, sometimes we have integral membrane proteins, and sometimes we have peripheral membrane proteins. I'm not going to ask that you know the difference in all of these, but just know that there are proteins that help substances move in and out. So here is another diagram for you. You can see the cholesterol molecules. Remember, those are important for stability and fluidity. They help to increase stability and fluidity. And here are some proteins. Remember, those are going to help substances move in and out of the cell. All right, so let's talk about membrane transport. So a cell's plasma membrane is what we call selectively permeable. And basically that means that some substances get to come straight through and other substances have to go through those protein channels, which we've already looked at. So here you can see the protein channels here. There are two major types of membrane transport. We have passive transport and active transport. The major difference between these two is passive transport does not require any energy, so no ATP is needed to move molecules inside or outside the cell. In active transport, ATP is needed to actually move the molecules inside or outside the cell. All right, so now I want to talk even more in depth about these two different transport mechanisms. So here with passive transport, we have two different mechanisms. We have diffusion and we have osmosis. In active transport, we have endocytosis, exocytosis, and pumps. And basically, endocytosis means that we are going to bring substances within. So endo means within the cell, and exo means that we are going to excrete them from the cell. And you can actually see examples of endocytosis here. We can form those little vesicles to bring them in. And then exocytosis, you can see that those little vesicles form and they are ejected. The material here is ejected from the cell. And then lastly, we have pumps. There are really important pumps in your body. One of those is the sodium-potassium pump. And the sodium-potassium pump is going to be really important in muscle contraction. So diffusion, I want to go back to passive transport. Diffusion is the tendency of molecules to move from high concentrations to low concentrations. And when I make brackets, that means concentration. So diffusion is the tendency of molecules to move from areas of high concentration to low concentration. And let's look more in depth at that. And we'll come back to osmosis. All right, so diffusion. Remember, it's the tendency of molecules to move from high concentrations to low concentrations. I like to think about it as like creamer in your coffee. You can also see in this example, we go from a high concentration of the molecules here in the dropper, and we put them into the water, and they begin to diffuse or spread out until here they've reached equilibrium. And this is what happens if you go to Starbucks in the morning or wherever you may go to get coffee, and you ask for creamer. And if you pour the creamer in, it will disperse throughout the cup of coffee, and your coffee will go from dark black or dark brown to that light brown color, depending on how much creamer you use. I don't drink coffee just because I would have my coffee with my creamer. So yeah, anyways. Let's talk about the rate and direction of diffusion. So there's a few things that can determine the rate and direction of diffusion. So remember, the direction is always high to low, and we're trying to reach equilibrium here. That's going to be our concentration gradient. Let's see. There we go. The size of the molecules is going to determine how fast they can diffuse. So small molecules are going to move much faster. And temperature is also going to affect the rate of diffusion. So it increases at higher temperatures. So diffusion increases at higher temperatures. I always use the example, if you have ever eaten at Chipotle, you know what I'm talking about. Basically, when you eat at Chipotle, sometimes the next day, or even sometimes, like, that same day, your stomach starts to make some weird noises. It's really your intestines there, but anyways, and you start to not feel so good, and you kind of get gassy, right? And so let's say you and your friend are in Alaska and you've bundled up, right? So you've bundled up and there's really no need for heat in the car, but it's wintertime in Alaska. And there's no need for heat because if you turn the heat on, you'll have to just take all those layers off. So you just stay bundled up, but you don't turn the heat on in the car. And you're going down the road with your friend after you've eaten Chipotle, several hours after, and you're not feeling so good. And you don't want to tell them what's going on, but you're feeling really gassy, and you just pass some gas, right? So you pass some gas, and you're hoping that your friend does not notice, right? You're hoping your friend smells nothing. It's gonna take your friend some time to notice. So maybe within that time you can be like, hey, let's pull over, or distract them somehow, or decide that on a nice cold winter's day in Alaska, it's a good idea to roll down the window. But if it's cold, the rate of those gas molecules, they're going to be slower to reach your nose, and so in your olfactory system. So anyways, let's take the same scenario and let's put us in, I don't know, Arizona, another A state, and it's really hot. Your air conditioner's broken, so the car, it's like really, really hot, and you're just driving a short distance. But anyways, same thing happens. You pass gas. Your friend is going to know right away, okay, what you have done, and that they probably need to pull over and let you handle that situation really quickly. They're probably going to roll down their window pretty fast because they're going to be able to pick up on the fact that you have passed gas, because the rate of diffusion increases at higher temperatures. And if it is really hot in that car, that person is going to smell that you have passed gas very quickly. Another example would be at Starbucks, you get the hot coffee, right? So you get the hot cup of coffee, you pour the creamer in, it disperses. Do you even have to have a stir to stir it? Not really, not unless you use sugar, because it should be hot enough that diffusion happens quite quickly and you don't even need to stir the coffee. But if you ever get served a cup of hot coffee and it doesn't, then you should tell them it's not hot enough. Also, if you've ever gotten iced coffee before and you try to add creamer, the creamer just kind of like sits on the top, right? It doesn't like diffuse through it. And it's really actually hard to get the creamer to diffuse through it because it is much, much, much colder. And then, is the membrane permeable to the molecules? That's another important part of diffusion. All right, so let's talk about the two mechanisms of diffusion. You have simple diffusion. Simple diffusion is going to be of nonpolar molecules. Lipids, for example, CO2, like gases such as CO2 and oxygen. And this is the example here of simple diffusion, where these molecules get to go on through the cell membrane, because remember, those fatty acid tails are nonpolar, and so are the molecules that are trying to pass through, such as lipids, and then you have CO2 and oxygen that can also pass through freely. They do not need the help of protein channels. Facilitated diffusion, you need transport proteins like these here. And that's because these molecules are going to be polar, so they're not like the cell membrane, which is nonpolar. So because they're polar, such as water, soluble molecules, an example of that would be glucose, also ions, sodium, potassium. Those would both be examples of substances that would need to come through into the cell via facilitated diffusion. So my example here for this, or analogy, is immigration. So my fiance and I do some traveling, and usually when I go to visit him in the UK, we both come back from the last time it was Italy. So we were coming back into the UK, and when you get to the UK border, it says UK citizens, and it says all other passports. And so he gets to go straight up, because he's a UK citizen, he gets to go straight up to these little gates, and basically it's just a gate, and you put your passport down, and the passport is scanned, and the gate opens, and you get to just walk on through. No big deal. Very similar to simple diffusion. But I have to go through the all passports line, which is usually pretty long. And you get interrogated in that line. You get asked questions about how long you're staying, how much money did you bring with you, where do you work, where does he work, how did you meet, when did you meet, how long have you been dating, etc., etc. And so it's a much harder process, because you have to go up and you have to speak with somebody, and then they get to determine whether you get to come in or not. It is not, ooh, scan my passport, walk on through, no big deal, like it is for UK citizens. And that's because I'm not like the UK, right? I'm not from the UK. Being from the USA, I have to go through and talk to somebody and be questioned before I can enter. And so that's kind of what's happening here with facilitated diffusion. These molecules don't just get to come on through freely. They don't get to scan their passport and walk on in. They actually have to go in via these proteins here in the membrane. They can't just diffuse through. And that's because they're not like the membrane. They're polar. And the membrane is nonpolar. All right, so then we have osmosis. Osmosis is basically the diffusion of water, or the tendency of water to move from high concentrations to low concentrations. So it's the tendency of water to move from high concentrations to low concentrations. All right, so let's talk about tonicity. This drives the direction of osmosis, and it's really going to depend on how much stuff is dissolved in the water. There are 3 different types of osmotic solutions, okay? So you have what's called hypotonic solutions, hypertonic solutions, and you have isotonic solutions. So let's draw our cell here for all 3 of these. So a hypotonic solution describes the solution that's outside the cell. So the solution is made up of the solute and the solvent. So the solvent here is going to be water, and the solute could be glucose molecules, it could be salt molecules. I like to think of like salt, for example. I think that when I think solutes, I think like salt in the water. So here in a hypotonic solution, that means that there is less solute outside the cell in the solution. And if we have less solutes, we have more water. And inside the cell, we have the opposite. So we have more solute and less water. So if we put a cell into a hypotonic solution, the water is going to move from high to low, more water to less water here, right? So the water moves in. And what does that do to the cell? It's going to swell. And think of it as like the big O, hypotonic, the big O. It's going to get much bigger and swell if we put a cell in a hypotonic solution. In a hypertonic solution, we have the opposite. This is going to be a solution. This is like salt water, basically. It's going to be very high concentrations of salt in the water, or some other type of solute, glucose maybe. So you're going to have more solute and less water in the solution surrounding the cell. And inside the cell, you're going to have the opposite. So more water and less solute. And so here, remember in osmosis, water moves from high to low concentrations. So here, water is going to move out. And that's going to cause the cell to shrivel. And in an isotonic solution, the solute is equal to the H2O, solute inside the cell and outside the cell. So water is going to move freely. And this is a normal cell. So I think of it like this. If you have a potato, we use like little potatoes, and if the potato goes into a hypotonic solution, the water is going to move in and the potato is going to swell up because there's more water in the surrounding solution. If I take that same potato and I put it in a hypertonic solution, that means that there is less water in the solution and more solute. So the water that's inside that piece of potato is actually going to leave the potato, and the potato is going to shrivel up. I think of like a plant kind of shriveling up. And then if I place it in isotonic solution, the potato is neither going to shrivel nor swell. It's just the water is going to move freely because that's its normal solution. Iso also means same, so you can remember that too. Iso means same.